Let's begin with our lecture, and in the first section of our lecture we will discuss the etiology and pathogenesis of botulism. And when we will discuss the etiology, what are the different causes of botulism, how the botulism occurs, and pathogenesis, how the progression of the disease occur, we will discuss about what is botulism. That's important. We should know what is botulism, what causes botulism, what are the different stages through which the disease progress. So definition and overview of the botulism. Then we will discuss different types of botulism depending on the different serotypes of the botulism toxin, which is from A to G. And then we will also talk about the common for bacteria or clostridium which can cause the botulism. And then we will discuss also the botulism toxin and how it produces the effect which are very, very significantly related to the botulism. So let's first talk about the definition of botulism. What is botulism and how we define the condition known as botulism. So botulism is a neuroparalytic disease. So as we say, it's a neuroparalytic disease. That can even tell us that it's a serious condition because it's a neuroparalytic disease called paralysis of the nerves, and it causes serious manifestations. Later on we will talk about them. So it's a neuroparalytic disease caused by the toxin produced by the bacteria which is known as the botulinum toxin, one of the most toxic substance known. So it's a very, very serious condition. It's one of the most toxic substance is the botulinum toxin known, and it can cause the neuroparalytic disease. Three additional forms of botulism. They are caused by in situ toxin production, and then also by the spore formation and the germination of the spores produced by the clostridia. So it's a neuroparalytic disease caused by the production of the toxin known as botulinum toxin, and it is also by the in situ production of the toxin and by the germination of the spores. So all these factors contribute to the development of the disease process. Overview of botulism, symptoms reported in patients receiving. So if patient receive injection of botulinum toxin, since botulinum toxin which is produced as a result of infection, which is a neuroparalytic toxin, when it is injected to the patients, they produce the symptoms, and that can give us the idea what are the different manifestations of the botulinum toxin or the botulism. So it is injections of the botulinum toxin, symptoms reported in patients who receive this toxin. Now, types of botulism: wound botulism, infant botulism, and the adult intestinal colonization botulism. So these are three main types. We have wound botulism, infant botulism, and then it's in the adults. It caused by the colonization of the intestine by the botulism, botulinum, clostridium botulinum, that cause the disease. So three main types of the botulism are present. Etiology and pathogenesis. 7 serological distinct confirmed serotypes of botulism are present. So we have 7 distinct serotypes from A to G. So it's A, B, C, D, E, F, and G. And then different serotypes are more aggressive, more pathogenic as compared to the other. We will discuss about them later on. So we have 7 serotypes from A to G of the botulinum toxin. Botulinum toxin is produced by 4 species of Clostridia. We have 4 species of Clostridia that produce botulinum toxin. Botulinum toxin has 7 serotypes A to G that can produce different features. These 4 types of Clostridia, we have Clostridium botulinum, that's the most common Clostridium botulinum. Then we have Clostridium argentinense, Clostridium baratii, and then Clostridium butyricum. So these are 4 types of Clostridia that can produce botulinum toxin, which is a neuroparalytic toxin, one of the most severe form of toxin that is present or that is known. So it's a serious disease caused by the production of the toxin from these 4 main Clostridia, and then its toxin is subdivided into 7 subtypes or serotypes. It's an anaerobic, that's the Clostridia. So it's an anaerobic. Anaerobic are the microorganisms that do not require oxygen for their growth and multiplication. So they are anaerobic, gram-positive organism. They can stain by the gram stain. So they are gram-positive and they are anaerobic. Forming subterminal spores. They form these spores, and the microorganisms that form these spores, they are very resistant to destroy by the heat and temperatures, so they can grow and they proliferate. So they can produce these subterminal spores. Clostridium botulinum and Clostridium argentinense spores. Toxin production require, what are the different factors that play an important role in the production of botulinum toxin? Anaerobic environment is one, because these Clostridia grow and they are an anaerobic gram-positive organism, so they require an aerobic environment. Then they need pH of more than 4.6, low salt and sugar concentration, and they require temperature of more than 4 degrees Celsius. So these are some important factors that are important for the production of the toxin by these Clostridia. They have some pH requirement, they have temperature requirement, it should be anaerobic, and they should have low salt and sugar concentration. All these factors help in the proliferation of Clostridia and production of the toxin. Food-borne botulism. Food-borne, as the name show, is spread by the eating of contaminated food. So caused by the food which is contaminated with the botulinum toxin. If that contaminated food is ingested, it can cause the food-borne botulism. No confirmed host-specific factors are involved in the disease. So no specific host factors are involved means like low host immunity and some other like patients, some infections develop in patients who are using antibiotics or chemotherapeutic drugs or who have a low immune system or altered immune responses. So this is not related to botulism and it has no specific host immune factor associated with disease. If the food is contaminated with botulism toxin and the person consume that contaminated food, it will develop botulism. Wound botulism, so wound infected with the botulism due to contamination of wound with clostridium botulinum spores. So if spores are present and that wound is contaminated with the clostridium botulinum spores, that can cause wound botulism. Subsequent spore germination, so contaminated with spores, germination of spores occur and toxin production in anaerobic melee of abscess or wound. So contaminated with spores, germination of spores occur. These germinated spores produce botulinum toxin and then this toxin will produce different features or manifestations which we will talk later on. So that's how the wound botulism occur. Food-borne due to eating of contaminated food by the botulinum toxin, wound which are infected by the spores, germination of spores occur and those spores produce the toxin which later on produce the botulism. Infant botulism, infants from absorption of toxin produced in situ by toxigenic clostridia colonizing the intestine of children less than 1 year of age. So the toxin produced in situ, it is absorbed by the intestine of the infant which is less than 1 year of age. Colonization of intestine occurs because normal bowel microbiota not fully established. Normal bowel microbiota, normal bowel microorganism, which is the normal mechanism of protection that is not fully developed in infants, so they are colonized by the Clostridium bacteria. This theory is supported by the studies carried in animals, not in humans. Next, adult intestinal colonization botulism. Very rare form. Adult intestinal botulism is a rare form. Pathology is similar to infant botulism. Colonization of intestine occurs by the botulinum toxin. Occurs in adults with anatomic or functional bowel abnormality. So if there is any abnormality of the bowel anatomically or functional, it can cause the proliferation or colonization of the Clostridia. Or have recently used antibiotics. Antibiotics helping toxigenic Clostridia compete against normal microbiota. So as in children, we said that the normal intestinal microbiota is not fully developed. So when adults who are using antibiotics, it decreases the level of normal intestinal flora. That helps the botulism to grow and then it can cause the disease by it helps the competition between the botulinum, clostridia, and also the normal intestinal microbiota. Despite anti-toxin treatment, what occurs? The relapse due to intermittent intraluminal production of toxin. Even though botulinum antitoxin is used, it can cause recurrence of the disease because of the production of intraluminal toxin. May be observed in patients with adult intestinal colonization, botulism. Now, the botulinum toxin, which is producing all these effects or causing botulism, is the botulinum toxin produced by the clostridia. What is this botulinum toxin? It's a zinc endopeptidase protein of 150 kilodalton consisting of 100 kilodalton heavy chains and 50 light chains. So what is this toxin? It is zinc endopeptidase protein, which contains light chain and heavy chain. Heavy chains are more as compared to the light chains. Botulinum neurotoxin enters the vascular system. So after the toxin is produced by the Clostridia botulinum, it enters the vascular system and transported to peripheral cholinergic nerve terminals and neuromuscular junctions and peripheral ganglion, etc. So it enters the vascular system, it enters the neuromuscular junctions and peripheral ganglion, where it can insert its neuroparalytic effect and produce different manifestations. Now, the steps in the neurotoxin activity, how they perform this neurotoxin activity. First, the heavy chains present in the toxin, which is about 100 kilodaltons, heavy chain binding to the nerve terminal occurs. This causes internalization in endocytic vesicles of the heavy chains, internalization in endocytic vesicles, translocation of light chain to cytosol, and light chain serotype specific cleavage of one of several proteins involved in release of acetylcholine. So all these are the steps that leads to the neurotoxic, neuroparalytic effect caused by the botulinum toxin by attacking the nerve terminal, internalization, translocation to cytosol, and then cleavage of one of several proteins involved in release of acetylcholine. And this inhibition of acetylcholine released by the toxin cause characteristic flaccid paralysis. So this inhibition in the release of acetylcholine as a result of cleavage of the protein involved in the release of acetylcholine is involved in the flaccid paralysis, which is characteristic of botulism. Now, different serotypes, we said there are 7 from A to G of the botulinum toxin. So these serotypes, all of them are not equally pathogenic or damaging. Some are more as compared to others. So let's talk about the 3, 4, which plays more important role in the progression of the disease. We have serotype A, produced most severe syndrome. So serotype A is most severe, with greatest proportion of patients requiring mechanical ventilation. So they are severe, they cause paralysis of and the inhibition, ventilation is affected, respiration is affected, and these patients require mechanical ventilation. Type B is appear to cause mild disease than type A. In both food-borne and infant botulism, serotype B is involved. Next, serotype E, associated with foods of aquatic origin, produce syndrome of variable severity. And then we have rare cases of illnesses caused by serotype F, characterized by rapid progression to quadriplegia and respiratory failure, and also by relatively rapid recovery occur in this. So we have A, B, and then we have E and F. These are the serotypes. Serotype A is most severe, the patient needs mechanical ventilation. The other one are relatively less severe, and then serotype F cause quadriplegia. Quad means four, so all four limbs, upper two limbs and lower limbs, they are paralyzed, and it can cause respiratory failure. So it's these are to some extent are serious, but the serotype A cause more serious disease. Some serotypes can be differentiated into subtype through neurotoxin gene sequencing. So that concludes section 1. Thank you for watching sqadia.com.